The endomembrane system is a collection of organelles that are all linked to each other through membrane that basically flows from one organelle to the other. So in effect, most of the parts of it are actually connected. And if you look closely at the images of the cell or at the model in class when we meet, you'll see how the membranes actually just touch each other and flow into each other. So the endomembrane system includes the nucleus, the endoplasmic reticulum, or ER for short, the Golgi, lysosomes, vacuoles, and the peroxisome, and we'll go through each one of these now. It also includes the plasma membrane because the plasma membrane is going to be connected to these other organelles. Again, as I said, either they actually literally the membranes connect to each other, or they're going to pass between each other through structures called vesicles, and we'll see how that works in a little bit. So we'll start with the nucleus, and the nuclear envelope is part of the endomembrane system. It's a double membrane. That means it's actually 2 layers. It surrounds the genetic material. The outer membrane of the nucleus actually becomes basically the endoplasmic reticulum. And again, I encourage you to look at the models in class to see how that works. In the nuclear envelope, there's nuclear pores. These are openings that are going to be really important for allowing things in and out of the nucleus. In particular, later on in chapter 12, we'll see that RNA needs to be able to leave the nucleus. Inside of the nucleus, we have the genetic material. And remember, I said this is in physical units called chromosomes that are actually made of DNA and protein. The function of the nucleus is to really protect and organize the genetic material. And this makes sense if you think about eukaryotes needing it and prokaryotes not needing it. To put in perspective the amount of DNA, I'll just tell you that a bacterial cell has about 100 genes. A eukaryotic cell like our cells can have like 40,000 genes. So it's a huge amount more of DNA. And when you start to have more DNA like that, it gets messier, it gets more vulnerable, and so having it protected in the nucleus makes sense when you have so much DNA. And so that, again, is the primary function of the nucleus. Inside of the nucleus, we also have structures called the nucleolus, and it is where ribosomes are made. Even though it sounds like there's just one and the pictures in your textbook always show only one nucleolus, in reality there's lots of them. And I like to mention that because in the lab you'll notice that there's lots of dark spots within your nucleus, and that's because there's many nucleoli inside of the nucleus. And so you'll see those little dark spots again when we do the lab in class. So here's the nuclear envelope. Remember, it has 2 layers. There's pores through which materials can pass. This is a nucleolus, and again, it's a little dark spot that you can see in the microscope. And then this is the DNA. In this case, the DNA is loose. We'll see that at certain times in the cell's life, the DNA will be tighter and more visible. Okay, next part of the endomembrane system is the endoplasmic reticulum, or ER for short. It's a network of flattened membranes. So you can see it kind of looks like like you took a circle of membrane and squished it. And it's a network because it's all contiguous with each other. It all kind of flows and bumps into each other, and materials are going to pass between them. And again, I encourage you to look at the model to sort of see how that works. There's kind of 2 parts, the membrane part and then the dark part here in the image. That's the lumen, or what that basically means is interior, inside of space. There's 2 types of ER. There's rough ER and smooth ER. So back here is the rough ER. See those little bumps on there? That's what makes it rough. If you imagine touching it, it would feel rough. This is smooth ER. Notice it doesn't have those little bumps. If you imagine touching it, it would feel smooth. Those little bumps are ribosomes. So rough ER has ribosomes, smooth ER doesn't. This directly correlates to function. Rough ER is studded with ribosomes, so that those little, again, the rough part. And remember, we said earlier that ribosomes make proteins. So the function of rough ER is to make proteins. Specifically, it's going to make special proteins, or it's going to make proteins that have to end up in certain places in the cell, or that have to leave the cell through the secretory pathway. So protein synthesis and sorting to specific locations. The smooth ER does not have ribosomes, so it has no role in protein production. Its primary functions are detoxification. So, for example, alcohol is a material that the smooth ER detoxifies. It's involved in carbohydrate metabolism, so producing glycogen and things like that. Maintaining calcium balance, so the ER lumen will store calcium. And it's also responsible for making lipids and modifying them. So kind of a lot of sort of diverse functions in the smooth ER. But primary one being detoxification of specific molecules, and alcohol being a classic example. The Golgi apparatus, Golgi for short, Golgi body, Golgi complex, all of that's referring to the same thing. Looks really similar. It's also stacked, flattened membrane compartments, but they are not physically connected to the ER. So again, take a look at the model to see how it's a little bit separated from the ER. Its role is really to accept materials from the ER, but it's going to accept them in this form called vesicles. So I'm going to do a little bit of a, and you'll see the figure too from your text. So the ER is all this stuff and it kind of touches each other. My drawing here is not very pretty. And then you have the Golgi, which is a little bit separate from the ER. And there's sort of three parts to the ER that we'll see. There's the cis, trans, and the medial. So three distinct compartments. Materials are going to go to the Golgi and travel between the Golgi in these little bubbles, little bubbles called vesicles. And they are really just like little bubbles of membrane that contain materials. So they're going to have to travel from here to here. And so that's what connects the Golgi to the endomembrane system is vesicles. The Golgi has three functions: secretion, which is to secrete things to the outside of the cell or release them to the outside of the cell; processing, which means to modify materials; and sorting. So sorting means to send to its correct location. People often refer to the Golgi or use the analogy of a post office because its primary job is to accept materials, to read what destination they're supposed to go to, and then send them to that correct location. So to really see how the Golgi works, we're going to talk about the secretory pathway. So the secretory pathway is how proteins are secreted to the outside of the cell. So an example of this is like in your mouth, right? You have protein enzymes that are released into your mouth to digest the food as soon as you start chewing it. And those are made of protein, and they have to be released from the cells in your mouth, from your salivary glands, into your food. And so the cells in your salivary glands will make the proteins and then secrete them to the outside of the cell. Because this is proteins, they're going to be made in the rough ER, because remember that has ribosomes. So it's going to start in the rough ER. It will travel through the rough ER, and then eventually it will come out of the rough ER into a vesicle. So the protein starts in the rough ER, then it goes to a vesicle. From the vesicle, it's going to travel in the vesicle to one end of the Golgi. So it first goes to the cis Golgi. So where are these vesicles coming and going? Well, membrane is a lot like bubbles. I used that term earlier. If you think about bubbles back when you were a kid and blowing bubbles, you can take like a large amount, like think about the soapy water across the little wand, and you blow into it and it forms the bubble. Well, that's what happens here. This is that soapy film, and then it's going to expand and make this little bubble that will bud off. So it buds off of here, then travels here. And then again, like bubbles, it can fuse. So a bubble can fuse back with membrane, right? So this becomes part of the Golgi. So vesicles bud out of membrane and they fuse back with membrane because they're made out of membrane too. So the vesicle from the rough ER comes to the cyst Golgi, fuses with the cyst Golgi, and then the materials pass through the Golgi. So they'll pass from the cyst part of the Golgi to the medial part of the Golgi, and finally they'll come out at the other end, called the trans Golgi. In the process, the protein might get modified, but primarily it's getting read. I say it's kind of like a zip code. It's reading the zip code that's on the protein, and it's deciding that that protein needs to get sent, in the case of secretion, to the plasma membrane. So it will get packaged into another vesicle, and then that vesicle will go to the plasma membrane. So here's that vesicle containing the cargo. That vesicle fuses with the plasma membrane, so there's the vesicle fusing. And when it fuses with the plasma membrane, whatever's inside of it gets released to the outside of the cell. And again, that's the definition of the secretory pathway. So the secretory pathway is the rough ER to a vesicle, to the cyst Golgi, to the trans Golgi, another vesicle that then sends it to the plasma membrane so that it can be secreted to the outside of the cell. And again, that highlights the role of the Golgi as a sorter and processor of materials, the post office that makes sure that things end up in their correct destination by reading the address on them. Okay, so the lysosome is also part of the endomembrane system, not part of the secretory pathway anymore, so we're leaving that behind, but still part of the endomembrane system. As I mentioned in the first video, these are only present in animal cells, not present in plant cells. Lysosomes contain a bunch of enzymes called acid hydrolases. They get that name because they hydrolyze all kinds of different things. Remember, to hydrolyze means to break down. So they break down proteins, carbohydrates, nucleic acids, and lipids. So they can break down things that the cell brings in. So some cells will actually ingest materials and bring them to the inside of the cell. And then the lysosome would be responsible for breaking down that food molecule, or that large molecule. So single-celled organisms will do this because that's how they get food. Certain cells in our body, like our macrophages, which are part of our immune system, will do this. But a lot in multicellular organisms, not a lot of cells do that. But every cell is going to need the lysosome for recycling. So this is a second function down here, autophagy. So this is the way that the cell will recycle worn out materials. So autophagy literally means self-eating. And so what we can see happening here is this is an old organelle inside of the cell. So it's just kind of worn out. Organelles, just like a machine, are going to get worn out and not be functioning that well anymore. And so what will happen is that will get wrapped in membrane. So kind of like a vesicle, this is wrapping it in membrane, so it becomes, it's bigger than a vesicle, which is why it's called an autophagosome. And then this autophagosome is marking this for destruction, basically. So now the lysosome will fuse with it. So you get a lysosome and the autophagosome fusing, and then that brings in the enzymes that will break down this organelle and recycle its parts. So the cell is extremely efficient. It doesn't want to waste these macromolecules, so it's going to break them down and reuse them for something else. So the lysosome, again, is for breakdown of macromolecules. It can break down food particles that might be engulfed by the cell, or it's involved in autophagy, which is this recycling of cell parts, of its own cell parts. Okay, vacuoles are another sort of extension of the endomembrane system. The term vacuole really is just sort of a loose catch-all for a large membrane-bound body. So there's different types of vacuoles. Their functions can differ depending on the cell type and the environment. The vacuole that we're going to talk about the most is the central vacuole. It's the large membrane-wrapped space inside of a cell. It's inside of a plant cell, sorry, so they're unique to plant cells. And again, they're oftentimes the largest object in the cell that you can see. They store liquid, so they mainly store water and solutes like salt for the plant. And because they're filled with water, they're also important for support in the plant. So that central vacuole has to swell with water, and that's what makes the plant cell firm. If the central vacuole is low on water, because the plant's not getting enough water, the central vacuole shrinks and the whole cell shrinks, and that's what causes plants to wilt. So that's the central vacuole in plants. So even though central vacuole is unique to plants, vacuoles in general are also present in other cell types. So you can have a contractile vacuole in certain types of protists. These ones are kind of cool because they squeeze. So you can have it sort of fill with water, and then it squeezes all the water out. And that's one way that certain types of organisms deal with water balance. You can also have food vacuoles. This is when cells engulf large particles. They'll need to bring it into the cell by wrapping it in membrane, and then the food vacuole can fuse with the lysosome, kind of like what I was just talking about before with the autophagosome. So the food vacuole will wrap around the food. So if I imagine, let me grab my pen here. This is my cell, right? Here's my food particle. What will happen is that it will bring that food particle in. So I don't know what's going on with the pen. And then it will have this food particle wrapped in membrane, and then I have a lysosome, and these will come together and that will break down the food. Okay, so really similar to what we saw with autophagy. Okay, so peroxisomes are another part of the endomembrane system. They're really small organelle found in all eukaryotes. They are involved in the breakdown of a really specific set of molecules. Some of them you probably heard of, free radicals, and molecules that create free radicals. H2O2 is an example of one of those. So free radicals, hydrogen peroxide, these are things that are gotten rid of by the peroxisome. So the name obviously comes from hydrogen peroxide, but just to be clear, that's not the only thing it gets rid of. It gets rid of free radicals and other molecules too. So the way that it gets rid of hydrogen peroxide is to do this chemical reaction, and it's carried out by an enzyme in the peroxisome called catalase. So catalase will take hydrogen peroxide and break it down into water and oxygen gas. And so it takes something that is bad for the cell and makes things that are not a problem for the cell, water and oxygen gas. Again, this is only present in eukaryotes. And so this is the basis of using hydrogen peroxide to clean a cut. When you take hydrogen peroxide and put it on a cut, your cells will take that hydrogen peroxide and break it down into water and oxygen gas, and your cells are just fine. It doesn't hurt your cells. But bacteria can't do this. And so when I put hydrogen peroxide onto bacteria, they die, right? So when we see the fizzing, when you put hydrogen peroxide onto a cut, the fizzing is the oxygen gas produced by your cells. It is not bacteria dying, okay? And this is a really common misconception. Bacteria don't like go, oh, I'm melting, like the Wicked Witch of the West when they're dying. And so they don't fizz or anything like that. They just quietly die. The fizzing is your cells protecting themselves from the hydrogen peroxide. We're going to use catalase as a way to study chemical reactions. So just sort of put a star by catalase and remember that it's a really important enzyme for us. Okay, last part of the endomembrane system is the plasma membrane. And remember, we said the plasma membrane is something all cells have. It's this important boundary between the cell and its environment. It's connected to the endomembrane system by vesicles that travel to it from the Golgi and other places in the cell. So its primary job is to be a boundary and to allow things to move in and out of the cell. It's called being selectively permeable, so it's picky about what it lets in and out. It's also involved in cell signaling, so there's going to be molecules embedded in the plasma membrane that are going to allow the cell to communicate with its environment. Because it's the border, it's the only part that will come into direct contact with the environment. For that same reason, the cell membrane is also involved in adhesion. it is what will hold the cell in place. So if you think about like your skin, for example, it's layers of skin cells that are adhered to a basement membrane or a basic layer that holds the skin in place. The plasma membrane will get a whole chapter of its own. So chapter 5 is all about the plasma membrane and cell transport. We'll talk about cell signaling a little bit more in chapter 9 and cell adhesion a little bit more in chapter 10. So that is the entire endomembrane system. As you may have noticed, it covers almost all of the organelles that are in the cell. We'll talk about the last couple of organelles in the next video.